Thank you very much for those introductions. If I understood Raphael's Spanish, which I don't understand a lot, but you said we've known each other for some years. As by my count, that's more than 30 now. I still had brown hair. Okay, anyway, it's really nice to be here back again. I was here 2 years ago and gave a couple of lectures. The nice part about this to me is that our collaboration with Rafael Jimenez-Diaz and his program is now coming to fruition because I'm presenting some results as a direct collaboration with this project. So I'll be talking about Verticillium dhalie, the biology of it, in relation to verticillium will. And just not sure where I should stand here. Okay, I'll try over here. I know many of you may be familiar with Verticillium dhalie, but I just want to go through some of the basics for those of you who are not familiar with this fungus. And in a moment, I'll get to why we're interested in this and also where we're going and what sort of things. But let's just start here. This is a soil-borne fungus that's really famous as a plant pathogen. It has a really broad host range. It infects many different plants. It causes lots of problems. And it's a really difficult fungus disease to manage because the microsclerotia can survive in soil for many years. I guess apparently the record is up to about 15 years. So it makes management extremely difficult because once you can't just rotate crops and get rid of it. And also with rotations, some of the rotation crops are also susceptible. It's a very complicated system. And one of the things that's really necessary for managing disease is breeding for resistance to verticillium wilt. So now, some of these things will become more clear later what their total relevance is. The disease cycle looks like this. It starts with microsclerotia in the soil that germinate. They infect the tips of roots. The fungus then colonizes the cortex, enters into the xylem, and then starts colonizing the plant through the xylem. And eventually it causes chlorosis, wilting, and the fungus colonizes the tissue of the whole plant. And as the plant senesce and die, the microsclerotia are released back into the soil. Okay. Okay, so one of the things that will emerge throughout this talk is that this fungus has a clonal population structure. It's an asexual fungus. So all the variation is caused by mutation, or at least this is, well, I'll start out by saying this is kind of the model for where we have always thought of Verticillium dhalie. This fungus can be divided into 8 different vegetative compatibility groups. And you'll see this term several times, the VCGs, vegetative compatibility groups. And this is a useful marker for describing populations, but it also has very important biological significance, which we'll get to a little bit later. And that is that individuals in the same vegetative compatibility group can form stable heterokaryons. They're hyphae can fuse and they can share nuclei. And this is actually the definition for how VCGs are defined. And biologically that has some relevance in a moment. We'll get there. Okay, and we'll also come back to this figure in a little bit. But what I want to point out now is that with using different molecular markers, this is with multiple gene sequences and AFLPs, this is some work previously done in Raphael's program, there are the VCGs correlate well with the different molecular markers. So these are the different VCGs, and you can see there is a tight correlation between VCGs as a marker and molecular markers that define the different clades. The fact that different types of markers define the same clades is a really good indication that this is a clonal fungus, that there doesn't seem to be much recombination going on. But we'll come back to that. That will be a major theme of this talk in just a moment. Okay, so the interest in Verticillium dhalie in Spain in particular is because verticillium wilt is a serious disease of many different crops, but especially in olives and in cotton. And this is actually not just a problem in Spain, although that's the main focus for what we're talking about now. Certainly lots of other countries have verticillium wilt in lots of crops, and apparently in Turkey they've got a very similar situation with severe verticillium wilt in olives and cotton both. Okay, the reason that this has become a more serious problem in the last 30 or so years is because of the emergence of a defoliating pathotype. And here are some symptoms here. Here's an olive tree that's completely defoliated because of infection with a particular clone of the fungus that causes defoliation. So that's known as the defoliating pathotype. And all the defoliating pathotypes are in vegetative compatibility group 1A. So you'll see this over and over again, the VCG 1A, or sometimes just 1A. And just in terms of how this fungus gets around, it's important to note that it's dispersed by irrigation and also in infected cuttings. And this is of particular importance for management because of the way that olive trees are propagated. It's really easy if you have infected plant material to spread the fungus all around the country. And this is apparently what has gone on here with both irrigation and in the cuttings. Okay, so the focus of my talk today is on the population biology and genetic variation in Verticillium dhalii. But so why should we care about this? For one, knowing about the variation in virulence, especially the defoliating pathotype, is really important. It's important to know whether you have the defoliating pathotype present or not. The management of the non-defoliating types is simpler. Okay? For breeding for resistance, you need to know what strains of fungi to work with in breeding programs. If you breed for resistance to the non-defoliating types, you might not have a satisfactory result with the defoliating type. And this is something that I'm particularly interested in, is how did the defoliating pathotype arise? Where did it come from? How did it form? And will it happen again? That's probably the more important question. But understanding how it arose the first time might tell us something more. And then finally, and I'll touch on this later, is the development of diagnostic tools that will allow us to detect and quantify the defoliating pathotype is another motivation. Okay, so the main questions that I'll be talking about today are first about population structure, and particularly looking at recombination and whether the clonal lineages, or VCGs, which correlate with lineages, whether these arose by recombination. And I can tell you the bottom line already is yes, it looks like there was a lot of recombination that in the past. We'll get to the data in a minute. Where did the defoliating pathotype come from? It was first observed in North America, so was it introduced or did it arise independently in Spain and other places in Europe? That's the second question. The third one is whether we can develop diagnostic markers to detect the different clonal lineages, and especially for detecting the defoliating pathotype. Okay, and I'll spend most of the time on this question right here about recombination, and then decreasingly less time on these others. So don't panic when I get, after a long time, I get to these next ones, but they'll be shorter. So all of these studies have been done using data from genotyping by sequencing. And this is a method that I want to spend some time on explaining right now before I get into showing you the results. I want to go over a little bit of emphasis on this method. Okay, so the trend in population genetics now is to use single nucleotide polymorphisms as a main tool. This is the current technology. It's probably with the exception of maybe completely sequencing every genome, this is about, even with sequencing the entire genome, what you end up with is analyzing lots of SNPs, single nucleotide polymorphisms. And the method that we've used is called genotyping by sequencing. And it's actually very similar to another method called RAD-seq, or restriction-associated DNA sequencing. These are all details. If you're familiar with RAD-seq, this might make some sense. Anyway, it's very similar to this method. It has a few key differences. One is it's about half the price. So, and the rationale is that with this method, we can get large numbers of SNPs distributed throughout the genome and it's a lot easier to do, faster, cheaper than whole genome sequencing. It's a way to reduce the complexity of the genome and still get a large amount of data. It was originally developed for, or this particular method was originally developed for looking at the maize genome, which is notoriously large and variable. So sequencing is not feasible. Okay, so there are 5 steps. I'm going to show you some figures of these in a moment, but I'll just mention them right now. Is that it starts with digesting genomic DNA with a restriction enzyme, ligating adapters to those restriction enzymes, pooling them together. They have barcoded sequences to identify them, doing Illumina sequencing, and then aligning the sequences to the genome, reference genome. So I'll just show you some cartoons of that now. So this is showing, as an example, 2 different genomes. It's just one segment of a chromosome, just to illustrate it. And these little brown spots are restriction sites for restriction digest. And you notice that this one right here is lacking a restriction site. This sample number 2 is lacking a restriction site here, which is a key part of this, because, well, you'll see in a moment. So you digest the genomic DNA with the restriction enzyme, ligate adapters onto the ends of them, and each sample has a unique barcode. You pool the fragments together, and then conduct PCR and sequencing, and then you align the sequences to the reference genome, and you can determine. So, for example, where this one, sample 2, is lacking a restriction site, there are no data at this site. So this is one of the drawbacks of this kind of method, but it does seem to work. So you take these millions of reads from the high throughput sequencing like Illumina, align them all to the reference genome, and then you can, every once in a while, you find polymorphisms like this. Okay, so this is just the method of getting huge amounts of data. And there are all sorts of filters and criteria for filtering out which signals are really SNPs and which ones are noise. So for example, when you see these variants down here, these are considered just sequencing error and they're ignored. When you get the same two nucleotides multiple times, then it's considered a reliable marker. And the bioinformatics part of this is actually done. Actually, the people that I contract with for the genotyping by sequencing present me with a file that's already been filtered, so I don't have to deal with much of the raw data in bioinformatics. Okay, so with verticillium dhalie, there is a reference genome that's been published and it's publicly available online. This is just showing the eight different chromosomes that have been identified, plus some of the contigs that have not been assigned to chromosomes. And I'll just show you, we'll just blow up this chromosome 8 just to show you what it looks like in a little more detail. So here's chromosome 8. The super contigs just are sequences that from the sequencing project that's been able to be assembled and located on chromosomes. So for our purposes, the important part is that we can align our sequences to this reference genome and know exactly where the SNPs are on each chromosome. Okay, so these are called super contigs or scaffolds. I will just be referring to them as contigs in the next slide, or the next view. Okay, so before I move on to showing you some data that we've got, some of the results, I just wanted to discuss briefly some of the advantages of this method. It's relatively easy. In fact, it's incredibly easy because someone else does the work. Can't beat it. We provide a microgram of purified DNA, and we get back data. So this service is commercially available at Cornell. So I just walk across the street with my samples, but anybody can contract with them, just mailing them samples. It's much cheaper than genome sequencing. You don't get as much data, but it's much cheaper. And it has the advantage of simultaneously discovering SNPs and doing genotyping. So this is a way of avoiding ascertainment bias, which is a problem in some studies. If you choose a small handful of SNPs because of detecting them with a small number of isolates and a small number of individuals, it's possible that you could really bias the results when you take it to another population. Anyway, this is one of the problems in population genetics. The bottom line, the bottom line is that we get thousands of SNPs, and they're distributed all through the genome. So here's what we've done so far. So the data I'm reporting today are on 141 isolates that were genotyped, and we have what we consider as reliable more than 26,000 SNPs. And it's actually a lot more than you need for most purposes, but it's okay. It's nice having that much. The SNPs. The number of SNPs that we find correlate really well with the size of each contig. So let me just show you these data. This is showing the number of SNPs per contig plotted against the size of the contig, and it correlates extremely well. So the conclusion that we draw from that is that the SNPs are pretty well evenly distributed throughout the genome. And this is nice to know, because then using this, we feel like we're getting a very good representation of the entire genome instead of just small pieces. Okay, so now let me move on to describing population structure and recombination. And to do this, I'm going to go back a bit to the clonal structure that's been shown previously. Okay, and really what we want to talk about is this fungus really asexual? Because this has been, it's been always thought of as being a classic asexual fungus because there's no known sexual stage. And this is true of many fungi. And verticillium, the entire genus of verticillium has been considered this way for a long time. The populations comprise distinct clonal lineages, which I alluded to earlier, but I'm going to show you a little bit more now, a little more detail. And these correlate with the VCGs. Okay. The problem with this is that strictly asexual organisms are not supposed to persist. If organisms are strictly asexual, they will accumulate deleterious mutations and eventually go extinct. So currently, the theory is that asexual organisms are either recently evolved, they've recently lost the ability to reproduce sexually, or they actually do experience some sex and we don't know about it. Okay, and I think this is kind of, there are very few exceptions to this rule. Okay, so one of the ways that we're going to detect recombination is to look at the incongruence of trees that are phylogenetic trees that are inferred from different parts of the genome. For example, from different genes. And if you have a strictly clonal or asexual organism, all of the genealogies from different genes should all match because for a very long time, those genes have evolved with the same evolutionary history because there's been no recombination between them. In contrast with recombining populations or sexual populations, if you have, this is showing the overlapping of phylogeny, genealogies of 3 different genes, there's no correlation among them. And if you look at you create a consensus tree in a phylogenetic analysis, there's no resolution here. These work perfectly for the clonal populations. Where it's recombining, you get no resolution, long branches and no resolution. Okay, so keep this in mind because this is something that, well, you'll see some evidence from recombination from this again. Okay, so this is the picture I just showed you before that was done with multiple gene sequences and AFLPs showing the correlation of vegetative compatibility groups to the molecular markers. Okay, and there are 2 major clades that have been shown here. This is clade 1 and clade 2 down here. And the 2 different markers match pretty well. So this is all evidence of a clonal population structure. Okay, but there are some discrepancies that have been shown, and I'll point out a couple here. First of all, the clades in VCG2b show up. There's this one subclade here that's called 2b334, and this one is 824, based on some other diagnostic markers. But they're in different clades, but the same vegetative compatibility group. So the question is whether these vegetative compatibility groups are evolved independently to the same vegetative compatibility group or whether they occurred by recombination. And the genetics of vegetative incompatibility groups are not known in this fungus, but in other fungi, it's controlled by multiple genes. So it's more likely, we don't know, but it's more likely that they arose by recombination than by convergence of mutations to the same genotype. Okay, the other thing to point out here is that these two different groups, there's VCG 2b and 4a in this cluster, and here it's 2a and 4b in this cluster. In other words, the 2s and the 4s group here, and the 2s and the 4s group here. The a's and the b's are just to indicate that there's some partial compatibility between 2a and 2b and 4a and 4b. Anyway, it's a little confusing, but it's likely that the 2a and 2b have a common evolutionary history before they developed a partial incompatibility. That's kind of the idea. The point with this is that we don't know whether these patterns evolved by recombination or by convergent evolution. And that's what that is one of the motivations for looking more closely at recombination. Okay, and more recently, and this is a paper that's just in press right now, again from Rafael's group, and also with Maria Del Mar Jimenez Gasco at Penn State University in the US. They looked at IGS, sequences of the intergenic spacer in ribosomal RNA genes, and also SNPs from 6 different polymorphic sequences. And these are the inferred phylogenies with the different VCGs listed. And every line that goes between these two different sets indicates some kind of a discrepancy between the two different marker sets. And this is what you would expect. You'd expect them to be completely congruent if everything is clonal, but these lines are indicating all sorts of swapping of different places in the phylogeny. So it's also, again, introducing the idea that maybe recombination is going on, or has gone on. Okay, so does this fungus reproduce sexually? And the evidence against it, just to summarize, is that there's no known sexual stage. Populations seem to be clonal, although it's not perfect. The mating type ratios are highly skewed. Almost every isolate that's ever been looked at is mating type 2. Mating type 1 is extremely rare. And mating can only occur between individuals of opposite mating type. So this highly skewed ratio would suggest that it's not reproducing sexually. Okay, in contrast, there are these discrepancies that I talked about earlier in the phylogenetic relationships. There's also some evidence using microsatellite genotyping of some populations in California that suggests that there may be some recombination there. That's a little bit hard to interpret for various reasons I won't go into, but it does suggest that there might be. And then finally, there's a kind of mutation, the repeat-induced point mutations. There's some evidence of this from some transposable elements. And this is a kind of a process that occurs in fungi only during meiosis. So again, this might be some evidence that sex is going on. Okay, so we don't know if it's reproducing sexually or not. So let's see what we can do. So these are the specific questions that we're going to address. Did the clonal lineages as identified by VCGs, did they arise by recombination? And if recombination did it occur by sexual reproduction or parasexual reproduction? And then finally, is it historical or is it something that's currently ongoing? So I'll address all three of these right now. Okay, with the 26,000 SNPs, we ended up with I created a neighbor joining tree just as a way of visualizing these relationships. I'm not trying to make any specific phylogenetic inferences. It's just a way to organize the data. But you can see that all of the VCGs, the same VCGs that were found previously, are actually shown in this tree also. So for example, here's clade 1. This is one subclade, and here's another subclade. So we get exactly the same patterns that were found from AFLPs and some a small sampling of SNPs. Okay, so it looks like it's just a clonal. It's the same story basically with 26,000 SNPs. So was there recombination? And here's where it starts getting interesting. What I didn't point out in that last figure was this clade right here. This is a whole new one that hasn't been seen before. It sure looks like it could be a recombinant between clade 1 and clade 2. And then we also get a smattering, a few other indications there might be recombination. This haplotype right here is intermediate between here and here, and the same thing here. These, and then this one. This is actually VCG6, which I didn't point out earlier, but that also looks like a recombinant. So the question is, how can we test for recombination? And what we've done is to use this program called RDP3. RDP is Recombination Detection Program is what it stands for. Okay. And what this does is, let me just walk you really slowly through how they're detecting recombination. It's, so if you had two different haplotypes, in this case, two different haploid parents, and you cross them. So, for example, this could be crossing over going on in on a homologous chromosome, and I've only shown the SNPs here between those two. What you'd expect to see is recombinant haplotypes that look like this. Okay, so they're just combinations depending on where the crossing over occurs. Okay, so if you look at this from the perspective of comparing the parents to a recombinant, you have, this can be done by looking at triplets of haplotypes, sets of 3 haplotypes. And what RDP does is it examines all possible sets of 3 haplotypes, or triplets. So here's an example right from the one that I just showed you in the previous slide. And what you could infer from this is that, looks like there's recombination going on just looking at the patterns of which nucleotides are shared with one haplotype versus the other. Okay, so in looking at some real data, what I'm showing here is VCG1A and VCG2B in the different colors. And here's a putative recombinant where it's sharing the nucleotides of the 2 different potential parents. Okay, so RDP3 does this with not just a small number of SNPs, but it takes the whole 26,000 SNPs and does this across the whole genome. Okay. And what it does is it looks at the similarities. For example, here's a recombinant type, and there are 2 potential parental types, and it makes a plot using a sort of a sliding window across the genome. And it will show the similarity with one parent, similarity with another parent, and this one across the bottom just shows that the 2 parents are quite different. There's very low similarity. Okay. So what it does is it tests the null hypothesis of whether this sort of pattern could have occurred by mutation or recombination. It looks for phylogenetic inconsistencies in different parts of the genome, depending on how much recombination has gone on. And it can, using these sorts of methods, it can identify where each unique recombination event that's gone on. And that's an important thing, because if you have different haplotypes that are related, it could be that they have the same recombination event. And you don't want to score that for every possible haplotype. You have to compare them all together. Anyway, it's a confusing process. And there's some recombination events you can't detect anymore because they get overprinted by other recombination events. And it's really a complex world here. Okay, and the other thing that can be done is that you can identify recombination breakpoints and hotspots, but I won't be talking about any of that today. We're taking a more gross view of the whole genome. Okay, so this is what we found. So far, we found more than 400 unique recombination events from our 26,000 SNPs. And like the number of SNPs and contig size, we get a good correlation between the number of recombination events and the size of the contigs. So here's number of recombination events per contig is plotted against the size of the contig. So like we did with SNPs, we're concluding that recombination is evenly distributed throughout the genome. Okay. So let's take a look at where these recombination events occur by clades. Okay, so in total we found 443 recombination events. Now, I'm going to try to walk through this slide slowly. I get confused with it myself, so I hope I can explain it to you. It's a little bit confusing. So we got 443 recombination events when we looked at the entire data set. If we remove the haplotypes that make up this clade right here, we end up with 364 recombination events. So the reasoning that I've used is that if you subtract that from the total, then 79 recombination events are involved in the formation of this clade right here. Okay? So I'm going to use this reasoning now as we sort of march through the different hierarchies of this cladogram. Okay. In clade 1, just looking at this clade, we found only 22 recombination events. And if we look only in VCGs 1A and 1B, right here, we get 2 recombination events. So the inference to me that I want to make is that 20 recombination events have occurred between this VCG and this one. All right? Is this convincing? I don't know if it is or not. This is how I'm thinking about it right now. Okay, and all of these recombination events in clade 1 are associated with this one haplotype. If you take this one haplotype out, there's no recombination evident in VCG, in this clade. Okay, now let's move down to this clade. Okay, we found 65 recombination events here and 90 here. And if you subtract that from 338, there's 183 associated with recombination between these two. And then moving in a little closer, there's 22 recombination events associated with VCG2A. All of them are associated with 3 different haplotypes right here. Okay, so I think this answers our first question of whether the the VCG's clonal lineages have arisen by recombination. I'm convinced that there's a lot of evidence. Oh, what I didn't get to, there's one more. Ah, yes. The recombination down here is associated with VCG6, which is this one, and this one haplotype right here. So I'm convinced that there's a lot of evidence of recombination, and that these, what we're calling clonal lineages, originally arose by recombination. So that's, I'll go out on a limb and make that conclusion. Okay, so now the question is, did recombination occur by sexual reproduction and meiosis, or did it occur by parasexuality? And to address this, we have to go through how parasexuality works in fungi. Okay, it's and on this side, I'm showing what happens during sexual reproduction of Ascomycetes fungi, and over here, this is the sort of the standard parasexual model that's been worked out in model organisms in the laboratory for the last 60 years or so. Okay, so sexual reproduction in fungi starts with plasmogamy of, in this case, a spore that acts like a male gamete, and a receptive hypha that then is like the female gamete. Okay, in parasexuality, this occurs by the fusion of hyphae that are in the same vegetative compatibility group. So here we go back to the same, this is an important part of parasexuality. They have to be able to form stable heterokaryons where they fuse and can share nuclei. Okay, so this is an important condition for parasexuality to occur. They have to be able to form stable heterokaryons. Okay, the next step in both sexual reproduction and parasexual reproduction is the karyogamy, or the fusion of the nuclei to form a diploid nucleus. And then there's a reduction from diploids to haploids. In sexual reproduction, this occurs in meiosis. In the parasexual cycle, it occurs by the loss of chromosomes during mitosis. How exactly this happens is, how it finally gets back to the right number, I think, is a matter of chance. But it's probably by non-disjunction of chromosomes during mitosis. And then finally, the other thing that occurs here is that there can be mitotic crossing over. So mitotic crossing over is actually a fairly rare event compared to crossing over that occurs during meiosis. And this is a key part of our interpretation. And then finally, you get recombinant ascospores in sex, and you get the occasional recombinant spores by parasexuality. Okay, so in parasexual recombination, the important thing to think about is that it only can occur within vegetative compatibility groups, not between groups. And there's very few crossing over within a chromosome with mitotic crossing over. So, in fact, with studies that have been done in model organisms, it's typically one crossover per genome or less in parasexual cycles. And then with meiosis, you get many different crossovers per genome. Okay, so let's look at the evidence here. What do we see for recombination within VCGs? There are actually only two places in this VCG and down in here where we have recombination detected. In this one, out of the two, none of them were none of the events were within chromosomes but there's only 2 events that were detected, so it's not a great surprise. Down here, though, most of the recombination events occurred in 7 of the 8 chromosomes. So this is not something that you would expect to see as a result of parasexuality, unless parasexuality happened multiple times, over and over and over again, which is not a very parsimonious explanation. I think it's more likely that it occurred by sex. Okay, what about when did recombination occur? What would we expect to see if recombination occurred recently versus occurred historically a long time ago? And I haven't got a really solid answer for this yet. I'm still trying to think this through. But think about this example again. Here are one parent lineage, the recombinant, and another. So obviously, if each clonal lineage starts from a recombination event, eventually what you'd see is more SNPs occurring within a VCG caused by mutation. So these SNPs, if they arose by mutation after recombination, they can be used to date the divergence time. And so let's take a look at this. The other way to think of this is look at non-recombining clades. If I take out all of the haplotypes that give us recombination and then look at the diversity of the nucleotides, what we end up is a pattern like this. And so here's the nucleotide diversity. These are actually fairly low diversities compared to what you'd expect across a whole species. And I'm not quite sure how to interpret them yet. I don't put any significance on these down here because we had only 2 haplotypes for each of these 2 clades. VCG2b, or clade 2b824, has the largest diversity, 3 or 4 times any of the others. Maybe this is an ancestral type. I don't know. Anyway, I'm not quite sure where this goes. The other problem is that comparing differences in diversity could be confounded with selection for different types. These are mainly agricultural. They're all collected from agricultural crops. And then also the sampling. Some of these were sampled widely. This was 77 individuals here. They were sampled from throughout the North America, Europe, Middle East, and China, whereas others, they're smaller samples. Anyway, the problem is interpreting this, like many problems in population genetics, it's quite difficult to give much of a definitive answer. My intuition is that recombination is not ongoing because we don't see very many recombinants, and probably this represents divergence time in the past. Don't ask me for a date. Okay, let's move on to, and as I promised, I'll spend a lot less time on these last 2 objectives. So, let's move on to looking at the origin of the defoliating pathotype. Okay, and so the defoliating pathotype was historically, it was first reported from California in the 1960s, which then prompted some searches for other defoliating pathotypes in other places. So not only was it found in California, but also in Arizona. And I just learned this morning it was found maybe in Peru. Is that right? Peru? So we don't really know where it's from. But the question that we're interested in is whether it was introduced into Europe from North America or whether this could have arisen independently. So the approach that we take to that is to reconstruct the genealogy of the defoliating pathotypes, which I've signified this as 1A/D because all of the defoliating pathotypes are in VCG 1A or clade 1A. Okay? So we want to reconstruct the genealogy. My goodness. I'll learn how to do this yet. And then overlay the geographic origins of each of the haplotypes that we find. So it's just a way of matching up geography to the phylogenetics. Okay, so this is what we found. This is with a sample of only 51 isolates from 51 isolates in the defoliating pathotype. And this, again, this is with the using the same SNP data set. There's a lot fewer SNPs in this clade. As you recall, that on the larger neighbor joining tree I showed you, it looked pretty much like a straight line. But there are some SNPs in here. I think it's a couple of hundred or something. Maybe not that many. Okay, so we got the neighbor joint. Oh, it's 54 SNPs. I'm sorry, 54 SNPs within this group. If we exclude the data where there's any missing data, we have data for only 35 SNPs. And that gave us 12 unique haplotypes out of these 51 isolates. So those are all the 12 isolates. The circles are including all those with the same haplotype. Okay, and if we construct a haplotype network, which is just a way of representing these instead of in a tree-like fashion, but more in a network-like fashion, what we see is a good correlation between geography and haplotype. So, for example, these two haplotypes are only in Western Turkey. These are in Greece, Israel and Turkey, Central Turkey. This is all the U.S. isolates. And then Spain from olives are here, Spain and Israel, and then Israel. Okay, so there's a pretty good correlation with geography. Well, what does this all mean? If we take another approach and do a coalescent analysis to reconstruct the genealogy and to map the mutations that we observe, all the different SNPs, onto this genealogy, we end up with a tree that looks like this. And the interpretation of this goes like this. First of all, the H1 haplotype down here is considered the ancestral type because there are no mutations that are along its branch. Okay, so that's the inferred ancestral type. Which means, if that's the ancestral type, then there were 5 independent introductions into the Mediterranean basin. Okay, because all the rest of these are sampled from the Mediterranean. If we look at this from the haplotype network again, what we see is that there was 1 introduction into Spain and Israel, 1 into Western Turkey, 1 to Greece, 1 to Turkey and Israel, or Israel, and then some sharing between the two, and then 1 into Central Turkey. So I think we've answered the question here of whether the defoliating pathotype arose once or whether it arose multiple times. In this case, it looks fairly clear that it arose once and then was introduced to these other places. Okay, and then finally, I'm going to finish up with just saying a few words on this is mainly to talk about the potential for developing diagnostic markers. And remember, we especially want to be able to identify the defoliating pathotype so that we can use that as a diagnostic tool for managing disease. And we'll start here again with showing this neighbor joining tree. This is with the 26,000 SNPs. And all of these groups are really clearly well separated except for 1A and 1B. So we have a large number of SNPs to work from that are, it should be very easy to just pull diagnostic SNPs out of our data set to develop diagnostic tools. The challenge is going to be in trying to separate these two. So let's take a closer look at that. If we expand this clade out into just looking at these, what we see actually is that BCG1B really is quite distinct from the rest of 1A. These are all 1A, and this is 1B down here. So we do have markers that we can use that are different between 1A and 1B. So we're pretty confident that we should be able to develop SNP markers for that. And then finally, and this is something that we're discussing, things are always nice in the discussion stage, but we'd like to be able to take these SNPs and develop a method where we can do SNP genotyping to estimate the frequencies of different lineages using some high throughput sequencing techniques like Illumina, which will give us estimates of frequencies of different SNPs. And from there, we should be able to, from a pooled sample, for example, from soil or from plants, to be able to estimate not only the total quantity of verticillium present, but the genetic composition of that population. Okay, so to just summarize, we've been using genotyping by sequencing. As a very powerful method. It's given us a huge amount of data in a short time, not terribly expensive. It's it's a powerful tool for doing evolutionary genetics. And with these markers, we found that recombination is really quite common throughout the history of this fungus, even though it may not be ongoing now. And the recombination, I think we can rule out parasexual recombination as the process. And people have talked about parasexuality in this fungus for a long time. Demonstrating it in nature has not been done. In fact, that's true for almost any fungus. Demonstrating it in the laboratory is easy, but from the significance of it in nature is not done. And then finally, the defoliating pathotype seems to have arisen in North America and introduced 5 times into Europe, once into Spain and Israel. And then finally, we have lots of SNPs that are specific to each vegetative compatibility group lineage, and we can develop lots of diagnostic markers for the defoliating pathotype and also for each individual lineage. And this is the next phase of our research once we finish up on this part. So I'd like to acknowledge my co-workers on this project. Rafael Jimenez-Diaz has been the PI on this project, and we work together with Maria del Mar Jimenez-Gasco and Concepcion Olivares Garcia. Funding has come for the Vertigen project from the Ministerio de Economía y
Competitividad. Okay.
Thank you. And also from the European Union. I'd also like to point out that I had a lot of help from people with bioinformatics and also in conducting the genotyping by sequencing for us at Cornell. And with that, I'd like to close.
Thank you, Michael. Estoy seguro que Michael estará dispuesto a recibir vuestras preguntas con extremo placer y yo creo que ha dado una demostración clara de cómo los conceptos que teníamos adoptados de la información del pasado se pueden cambiar a medida que uno progresa con las nuevas tecnologías y dirigir bien las preguntas y buscar con interés las respuestas. Así que si tenéis interés en preguntar algo y no queréis dirigir las preguntas en inglés, hacerlas en español, que yo con mucho gusto las traduzco para él. Pero yo animo a que dirijáis las preguntas que tenéis. I just try to encourage them to address you any question, even if they talk in Spanish, I will translate it to you.
I have two questions. So it seems clear that there have been back introductions from the USA into Europe, and all the countries that you show, of course, produce cotton. So do you think the pathway was introduced through the seed, cotton seed, or has been demonstrated that can be transmitted by seed? Or?
Okay, so the question is whether verticillium was likely to have been introduced on cottonseed.
Yeah.
I don't know exactly, but it certainly, from my understanding, it was a problem on cotton before it became a problem on olive. So either in seed or in breeding material or.
Maybe I can help somewhere. No sé si la pregunta que ha dicho la doctora Blanca Anda es si esa profusión de introducciones en áreas geográficas tan diversas se pueden entender como consecuencia de intercambios de semillas, en particular de algodón. En la historia pasada, el concepto asumido es que la semilla de algodón no es un buen vehículo para dispersar verticillium dhalie, a menos que no sea bien desborrada, de manera que la semilla es bien tratada químicamente o mediante calor. Esa posibilidad de que la semilla sea el transporte parece que estaba desestimada. Sin embargo, ha habido una publicación reciente de unos fitopatólogos en Turquía que han publicado en fitoparasítica varias veces, más de una vez, donde ellos están mostrando la facilidad con que verticillium dhalie se puede transmitir en semilla de algodón. Lo que no puedo cualificar yo por el momento es cómo de bueno fue el proceso de desborrado y si ese tipo de información que han producido ellos contrasta suficientemente con la vieja escuela de entender que la semilla de algodón no era fácil en cuanto a vehículos de verticillium dhalie. Pero son los dos elementos más cercanos uno y alejados otro, y quizás las cosas que no son fáciles cuesta mucho demostrar, pero al final parece que eso puede iniciar una forma de entender el proceso de distribución del organismo con la información que Michael ha explicado en términos de entender la diversidad hasta ahora. I will explain to you later because I brought different things into the question.
Because the isolates from the USA, from 1A, what is the host that you... Cotton, that's cotton.
Yes, they were, right. 1A from the US was isolated from cotton. Yes. And some of those isolates, or most of them, are older isolates from 20 or 30 years ago.
Even more.
Even more, okay.
I wonder how, if you can date the age of in which, how old each is haplotype. It can be the possibility that from Turkey, maybe some material moved to the USA, and then from there it got spread or not.
So I think they were. So based on that, on the gene tree from the coalescent analysis, it looked like it was pretty clearly 5 separate introductions. So you're asking whether I can date the time of that occurred?
Yeah, because if you put the Americans in the center,
yeah.
But there is one that has diverged more than from Israel and Turkey, Spain, Israel.
But none of them from that analysis would allow me to distinguish between when those introductions occurred. Yeah, they all seem to have been. Yeah, they're obviously not obviously, but I think that there have been secondary introductions. For example, the haplotypes that occur in Spain and Spain and Israel. It was introduced into one of those countries and then to the other. It's not likely that exactly the, well, it could be exactly the same haplotype that's introduced in both places.
Can you distinguish between cotton and olive isolate in Spain? Do they belong to the same haplotype, or do we have more than one haplotype?
I'm pretty sure they're the same. No, wait a minute. There was a, well, that's right. There's one of those groups actually was only from olive. But then there was other groups that were from olive and cotton. So yes, there's, but I don't know how well sampled cotton was compared to olive. We have a lot more samples from olive.
De hecho, esa es una pregunta que está en el fondo de toda la curiosidad, porque la muestra de aislados que tenemos representa la población original de cuando apareció el defoliante en las marismas, y hemos podido recuperar un número de aislados que representan históricamente los que debieron ser los primeros, y tenemos la muestra que representa los últimos que hemos encontrado en la valle arriba del Guadalquivir, en Olivo. Así que la intención es tratar de elucidar si o no se pueden inferir de ellos, que son descendientes entre sí, y por consecuencia ha sido una distribución de los originales, o se ha podido originar de nuevo. Es algo que Michael tiene ahora en entre manos para ver si puede ser. No, está pendiente.
So, I think you just said that we have a much larger sample coming. Yes. Yes, okay.
Porque él ha mencionado datos de 141 aislados y hay casi el doble disponible. Y esos dobles todavía still working on.
Yeah.
So next seminar will be addressing that particular question. Next year, el año que viene volverá de nuevo con esta nueva información. ¿Alguna otra pregunta? Adelante.
If sexual recombination is occurring, how come we haven't seen the structures responsible?
Well, so the question is, if sexual reproduction is happening, why has no one been able to describe the structures? This is actually a good. It's a problem with many different fungi, actually. That there's the genetics look a whole lot like recombination is happening, but no one knows of the structures. There's several different answers to this. One is whether people are looking in the right places. The other is, and I'll give you an example with Aspergillus flavus. This is another fungus I work on, actually. The sexual stage was not discovered until about 2 or 3 years ago, when people started looking inside sclerotia. They found the ascospores inside the sclerotia. It looks just like a sclerotia. Okay, so it depends on what you look for. The other is that it takes very little recombination, very little sex to produce a population with a recombinant structure. So a little bit of recombination goes a long way. So you combine with a rare event with not a lot of systematic searching. People probably gave up looking a long time ago. And it goes unnoticed.
And also the concept of asexuality, also deep-rooted, that not many people were just considering that may be already occurring. So
that's right, and it's only been in the last decade or so that people have come around to start thinking, wait a minute, ancient asexual lineages do not persist. I mean, they're very, very rare. There are some rotifers that are ancient asexuals, and that's about it. Maybe some mycorrhizal fungi, but they just don't exist. They don't persist.
Javier.
Ahora más alto, no te I understand that all isolates your work came from planta, recovered directly from the disease plant, regardless the host. But my question is about the soil isolate that you can recover directly from the soil, and about how this recombination mechanism can have an influence on the loss of pathogenicity of such isolates in soil. Do we have any information about it?
Okay, no, the answer is we don't have any direct information. It's something that we've been thinking about. We had actually a long discussion over coffee this morning about isolating, the challenges of isolating verticillium from soil. It's pretty difficult to do. So most of the bias in the sampling has always been to sample from infected roots. And even with that, the bias is to sample from roots of crop plants and those showing symptoms. So there are verticillium isolates that are in other what we consider non-host plants not causing disease, and that's something that's being looked at. But whether recombination could cause a loss of pathogenicity, I don't know.
Probably I needed the biological test.
Oh, yeah. It would be nice if we could make crosses in the laboratory and make these sort of tests, but
ahí ya para completar
it's not possible.
Mike no tiene oportunidad de decirlo porque son detalles menores, pero realmente en la colección de aislados que él ha utilizado ha habido algunos, no muchos, por eso quizá la información pues no ha lugar a interpretarla. Del 4A y 4B, algunos de Estados Unidos sí procedían de suelo, pero eran tan bajo el número que no hay posibilidad de vincular ninguna interpretación a diferencia, pero en general, siendo tan pocos, tampoco se han desviado del tipo de conclusión que se alcanza con los que vienen de planta. I just mentioned to him that that's very low number of isolates from 4B and 4A came from soil.
Right.
But I mean, there are so low number that doesn't mean much in terms of interpretation. Pero eso es una pregunta que está colgando en la mente de muchos que están interesados en el tema.
I think that we can use the opportunity to assess a collection of soil isolates before they're there. I mean, you know what I mean?
Yeah. You have a collection of soil isolate.
The microscope remain along years in the soil. So yeah, and probably they have lost their pathogenicity, and it would be very interesting to get non-pathogenic isolates.
Yeah. I think that my understanding is that it's very difficult to isolate from soil, and this is why they typically are not sampled.
It's difficult to isolate it, and it's difficult to clean it from bacterial contaminants.
Right.
So take a lot of effort. ¿Más preguntas? Sobre todo de la gente joven, son las más inquietas, supuestamente. Bien, pues si no hay más preguntas, sí, adelante.
My question is a general question.
Puedes elevar un poco la voz, que no te oímos.
In your opinion, in a hypothetical scenario of higher temperatures, with the climate change, is there a possibility of appear a new, more virulent pathogen in the Guadalquivir Valley, for example, by recombination?
I don't know what effect temperature has on selecting for higher virulence. I don't think we know enough yet about that, or at least I don't know enough about that. Or we don't know whether higher temperatures would result in more recombination. I think there are just too many unknowns. It's possible, but I don't know. I mean, but verticillium dhalie is found in a lot of different climates. In fact, it's not just hot, dry climates like here, but also in wetter, more temperate climates. So it's a variable fungus. I don't know what climate change is likely to do for it.
Yo por comentar un aspecto, puesto que la palabra del cambio climático ha surgido, y por trasladar una información, I'm going to comment on the coffee leaf rust. Ha habido una comunicación reciente en plan de alarma de ataques devastadores en cafeto, en la zona de Chinapas, en México, en cultivados en las alturas, de manera que ha sido la temperatura baja que en el pasado parecía excluir que hubiera ataques severos de esta enfermedad, pues ahora ocurre que hasta los agricultores están desconsolados porque es algo sorpresivo. En otras palabras, la leyenda es que el mundo de la biología esconde tantas posibilidades y si el cambio climático puede ir hacia un lado para calentar o hacia otro para enfriar, la biología persiste en el curso de la vida porque se adapta y que debemos estar preparados. Esa es la forma. Gracias por traer ese punto. Sí, hay otra pregunta por ahí. Sí, dale.
Hi, Michael. So, I have a lot of information in my head and I don't know how to make the proper question.
No te oímos.
Okay.
Ahora.
I'm saying that I have a lot of information in my head and I don't know how to make a proper question. Can you hear me?
Yes.
So, I've been thinking about rigid isolates that's in the old literature scientific used to talk about. There used to be rigid isolates from one BCG that they were able to complete the parasexual cycle with isolate from other BCGs.
From other BCGs?
Yes. Like rigid isolates, they were called.
Oh, bridging isolates.
Yes.
Yeah.
So, as you said, the parasexual cycle provide a little recombination, right? So,
yes.
It doesn't seem that they support the diversity,
Right.
high diversity we have. But I was just curious, now that we think that some sexual recombination is occurring or has occurred in the past, how do you, just curious, how will you focus your work? Do you think it's really important to keep looking for the sexual recombinations and what kind of isolate do they do the sexual recombination with? What about the mathematical forms that we have found in some DNA? And I know it's a lot of things.
Okay, so let me start with the parasexual part of this, because so the observation of bridging isolates, I don't think really changes my conclusions too much, because when I look at the recombination that occurs between VCGs, well, either there are many different cross recombination events. This doesn't necessarily mean it happened all at one time in one cross. And this is the problem with trying to use a historical collection to address this question. I mean, you take a sample from nature and you've got a long history of recombination, not just one generation. So any of the inferences that I make about the number of recombinations to infer parasexuality or meiosis they're not strong arguments. But the number of recombination events is pretty well evenly distributed across all the different chromosomes. And again, I just think that that's not likely to occur with parasexuality. Nobody's been able to demonstrate parasexuality from any natural populations of fungi with any convincing data. Now I say that being the author of one of the few studies that I think hints at it in Cryphonectria parasitica, which is a sexually reproducing fungus. So who cares if it's doing parasexuality? doesn't matter. The point is that parasexuality is something everybody loves to talk about and say, well, there's variation in the population, it must be that there's parasexual recombination. Okay, but that's not really answering your question. That's just getting at this polemic of. I don't know how to take this further yet. I haven't thought it through about looking for sexual fertility or where recombination's occurring or when it could occur. I don't know how to go forward other than looking at what we see in the historical record in the genomes.
¿Alguna otra pregunta? Si no, vamos a dar por concluido el seminario y agradeciendo a Michael su esfuerzo en poner en orden sus ideas y trasladarlas en una forma que ha sido accesible para personas que pueden estar familiarizados con un tema tan complejo. Gracias.